I would like now to invite Maricel Marin-Kuan. She is Senior In Vitro Toxicologist Specialist at Nestlé. She'll be talking about assessing NIAS, non-intentionally added substances. Yeah? Yes. In food packaging, combining bioassays with chemical analysis. Maricel, the floor is yours. A warm round of applause for Maricel.
Yeah, thanks a lot for the invitation. I have to clarify that I will not talk about the strategy of Nestlé on the packaging, but I will talk about the solutions that we are working to assess the safety of packaging, like the bioassays, because I am in vitro toxicologist. So I hope it is fun for you, because I think it's very interesting. So what is the role of bioassays in the packaging safety? As you know, in absence of chemical identification of the packaging substances that are migrating into the food potentially, we need to assess the safety anyhow, and the bioassays have been recommended as a tool because we can assess if there is genotoxicity, endocrine activity, and therefore it's very important to assess also the bioassays that we can use. And these have been reported by ILSI Packaging Task Force in their best practices for risk assessment of NIAS in food content materials and articles. And it's very hard to... So the bioassays or the endpoints, the biological endpoints that have been recommended is genotoxicity, endocrine activity, and cytotoxicity. Why these endpoints? As we have already seen now, today, I was not here this morning, but I'm sure you discussed about the importance of the genetic damage. We cannot accept that there is mutagenicity in the packaging products or that could be migrated into the food. This has been very clearly stated by the EFSA scientific opinion. Because of the severity of the genetic damage, we cannot accept any mutagenic compound that could migrate into the food. So this is very important in terms of safety to assess the genetic damage. So there is no question about that. For the endocrine active substances we have discussed before, there is the safety by design. We need to assess the endocrine activity because this is a lot of interest on their regulation, but also on the safety of the consumers. And therefore, this is clear reasons why we have to apply bioassays to assess the safety of packaging materials. And we need to establish a very good bioassays tools in order to be sure that we are assessing what we want to assess, the genetic damage and endocrine activity in this case. So this is a little bit the approach that we are applying in order to assess the food content materials at Nestlé. Of course, we have different departments, the chemical department and the bioassays group, where we have to assess the novel material, because now we are going into the recyclable, but also the packaging material that we have already in the pipeline, and we need to assess at this safe. It's not enough to change, but is the new material safe? And therefore we have to apply, as it was mentioned before, this safety by design, where we not only apply or assess the final product, but also the raw materials that are going to be used for the manufacture to be sure that they are safe. And we need to assess the migration that could be going into the food. So that for that we have a special protocol in order to assess migration. And we can also assess through the extraction. Extraction, why? Because we can do a worst case scenario in order to be sure, if I do an extraction, what is all what I can extract from this packaging. Of course, we need to apply the bioassays and different chemical tools in order to identify the potential compounds. And the bioassays are really based to be able and to be sure that we are assessing the genetic damage, the endocrine activity, the chemical analysis to identify and semi-quantify the compounds that could migrate, and to target or the suspected or non-target screening in order to try to identify. And this is, we know that this is the more difficult part, because it's very difficult it's a forest of peaks that we will get, therefore it's very complicated. And with the generation of all this approach, we can try to prioritize, to take a decision before we manufacture, or when we have already a product that we want to assess the safety. But there are still some problems in the application of the bioassays. It's very important, I think it's very clever to recommend to use the bioassays. However, are there bioassays that are viable, the correct ones, in order to assess the safety of packaging? And for us, this was a very important question, because the bioassays, as they are with the multi-wells, for example, the 96 web plates, when we put the packaging migration samples, we can create matrix effect. We don't have the limit of detection that is necessary, because maybe the molecules are a very, very little dose, then we will not detect them, and we will say it is negative, but it's really negative. And for the endocrine activity, the same. We need to be sure that we are exposing at the correct dose, because if we have very low concentration of potential toxicological concern molecules, we will not expose at the correct dose in the cells. So we need to be sure that the bioassays that we will apply are the correct ones. So that's why we have implemented a new in vitro competence to analyze packaging samples using the high-performance in-layer chromatography, or HPTLC. And I think this was a very clever idea, because if the problem with the bioassays is that we can have a matrix effect, I would try to avoid this matrix effect. How I will do that? I want to fractionate my sample. And this is the silica plates in the HPTLC. It was known since years, but now they have improved it a lot. And we can do a chromatography and separate our migration samples or extraction samples of packaging, which will allow to really elucidate what are the compounds that are present in my migration samples. And then I will pour or couple this chemistry to my bioassay. So I will not separate them, I will put them together. And what I will get is that I can assess with my bioassays, instead of using 96 plates, I will use the plate to have genotoxicity test or endocrine activity. These tests already exist are commercially available, actually. The SOS-UMUC is a gene that is present in bacteria, and there is a kit to assess the genotoxicity. It's very well known in the environmental field, and it's very good correlation with the mutagenic test AIMS, which is the one that is recommended, and the yeast estrogen or androgen activity screening. This is a kit that exists to assess endocrine activity. So we can pour these bioassays on the plates, and we can see here the green signals is the signal that we have a genotoxicity. And here in the dark, we have an inhibition. And in the green, we have activation. That means that we are getting this genotoxic effect or with endocrine activity. And this is the proof that the method, it can work, and that we can quantify it, which is the effect of the packaging material. So, but it's very important, and a really very breakthrough solution is that, okay, yes, I will say there is genotoxicity or there is endocrine activity, but still don't know which is the molecule responsible of this activity. What I can do then is to recover the bioactive bond for the genotoxicity or for the endocrine activity, and I can try to identify which is the substance responsible of this genotoxic activity or endocrine activity. And this is what we really need to do. We need to identify the molecule in order to assess the safety. So this is a way to a little bit to come into the identification of the packaging substances. So let's see how we can apply that to the packaging with a case study. So what we did is that we took paper material that is commercially available. It's not an Nestlé product, but we use it for the example. And here, what we did is that we spike it with different compounds that we know that are mutagenic. So I spike my paper, and then I do my extraction. I apply my samples into the plate of the HPTLC. I did the chromatography, I put my bioassays, and then I see if there was a genotoxic response. And the answer was yes, there is a genotoxic response. Please keep in mind that genotoxicity doesn't mean mutagenicity. Genotoxicity can be repaired, while mutagenicity cannot. So we need to assess mutagenicity. So what we do is that we can recover the band and do the mutagenicity, and like that exclude mutagenicity. Or I try to identify the sample. So what is the result that we got? This is a spiking with a mutagenic compound that is a reference compound that is used very commonly in the mutagenic testing, the 4-NQO. And here is, I see that I induce the genotoxicity, the UMUC, and proving that the method is working. Then I recovered the bound, and I did the mutagenicity test with the MCMPF test, and I see that there was induction of mutagenicity, proving that I am able to recover and to confirm mutagenicity, and then I was able to identify chemically with HPTLC-coupled that the molecule is the one that I spiked. I did the same with the glycidol, which is a molecule that is reported to maybe potentially be present in packaging, to be sure that it's not only a potent mutagenic, but another one which is more weak that could be also detected. And we were able to detect also the mutagenicity, because this is possible to, it is positive for mutagenicity in absence and presence of metabolic activation. So we were able to detect both and to identify the compound. So what happened with our packaging material, the paper? So here we see that we observed a bioactive bond at this retention factor, and we were able to identify that there was a dihydroabiatic acid methyl ester. It was the molecule that was inducing genotoxicity, and then we did the concordance. We acquired the standard for this molecule, and we test the molecule pure with the extract of my packaging. And we were able to observe that the same peak, it was corresponding to this molecule, which confirmed that this molecule is really the one that is present in the packaging. And then we did the Ames test, and these molecules were negative. So they were genotoxic but non-mutagenic, so I'm excluding mutagenicity. And with this, I proved that the packaging material, there was a genotoxic compound. I was able to identify the compound, and it was negative. So this was the same with another compound that is present in the paper, and this is logic because the abietic acid in reality is something that is present in the trees, and it's a molecule present in the plant. So it's kind of logic that we find it there, and it's very well described, and they are genotoxic but not mutagenic. So with that, with the reference compounds and with the compounds that we identified in the paper, we were able to demonstrate that the proof of concept of this technology, it's a very good one in order to assess the packaging safety. And therefore, we are trying to set up a screening tool in order to assess all the batches or the packaging that we can receive or that we need to evaluate. And the idea is that when we will have new packaging material, or there is a proposal to use a new packaging material, we can test with this approach, and we can do the chromatography, we can do the chemical profiling. This is very, very interesting because we can then build libraries of all the type of packaging that we can have, and this is very, very, very helpful. So not only the bioassays, but the chemical profiling that we can get there. And here it's, for example, a set of samples, the 10 samples of packaging, where we apply the different bioassays for the chemical profiling, the genotoxicity, with and without metabolic activation, the androgenicity, the estrogenicity, agonistic and antagonistic. And then we see with all the packaging materials that we have, if there is a signal. For example, here we see that there is estrogenic activity, there is anti-androgenic activity, but there are many others that are clean. Like that, I can prioritize and concentrate and focus on the ones that are clean, and for the ones, can we mitigate or not, or we just refuse them, and we need to change if there is a molecule that was identified. So we are very, very quite sure that this is a very good progress into the assessment of the packaging safety, and we are applying this for our approach on the suitability of to assess packaging safety, because we have the adequate limit of detection. We can facilitate the identification of responsible factors, which is really key. And I think it's the first time that we can really demonstrate that we are able to identify the substance responsible of the genotoxic and endocrine activity. And we are aligned with the recommendations on the use of bioassays, and we are improving the bioassays that are needed to be used. And this is very promising, and we are publishing because the more we apply it, the different groups, the best it would be recognized in the future. And with that, I will finish. So we are applying the bioassays, the general, the chemical analysis and the risk assessment all together. Of course, this is not only the bioassays, it's a lot of people behind that. And the conclusion is that we are anchoring the HPTLC to the bioassays is very promising. We can facilitate the identification of compounds potentially responsible of toxicological concern. And in the case that we study, we were able to demonstrate that we can detect mutagenic compounds, as the case with 4-NQO and glycidol. And this will be very helpful if we want to apply the threshold of toxicological concern, because the limit of detection is really in accordance with the recommendation. So, and with that, I want to thank you for your attention. And this is our group working on the biodetection and the packaging safety. Thanks a lot.
Thank you. Thank you, Maricel. Okay, we have 1 question, 2 questions, 3 questions. Stop. 3 questions. Sorry, yep, we have a microphone. Yep, please, go ahead.
Thank you. Very interesting indeed that is being done in this scale, but I would like to know about your estrogen assay, because many, or at least in a long time ago, many of those yeast assays detected anti-estrogens as full agonists. So how do you test that?
For the anti-estrogenicity?
Yes.
Yeah, you are right. I'm struggling with anti-estrogenicity. The advantage with the HPTLC is that the YES test, it's much better. So we improved the limit of detection because the 96 well plates is not so good. Huh? Do you agree? So, and here we have a much better sensitivity, and the signals are really, we are really picogram sensitivity. But for the anti-estrogenicity, it's very difficult, but even with the 96 well plates, with the Calux method or others that we have. So we always confirm with the classical ones, and they are very tricky for them.
You
confirm
with the classical ones,
yes,
like Calux or
always.
MCF7?
Exactly.
What about the overshoot that you have when you use, excuse me, when you use if you detect phytoestrogens, for example?
Yes.
You happen to have that. Normally, the yeast and the Calux give an overshoot. Do you
So
find
this
that?
is so this happened with phytoestrogens. We have, yes, when you have plant-based testing, you have overinduction of the luciferase.
Okay.
But this, we know that is overinduction, but it's nothing to do with the potency.
Okay.
It's just an artifact.
Thank you.
You're welcome.
Do you believe the amount of time required to conduct these detection tests will decrease over time with newer technology and newer maybe scientific discoveries?
Sorry, I didn't get
Speed.
the speed of the testing.
Yeah.
This is the best because it's in 3 hours instead of 24 hours. The incubation with the yeast of the bacteria, we saved a lot of time, and in 1 day we can have the results. And there is an automation, and we can program the nights, and then you do the half a day, if you would like.
How long did it take to develop that program?
Sorry?
How long did it take to develop that program?
It took time to set the methodology, but now we published, so people can apply it.
Thank you.
You're welcome.
Okay, I'm really sorry. I'm going to have to put a cap on questions. So I'm going to take 2 more, and that really is it. And I'm going to favor those who haven't yet raised a question. I'm sorry. So there's a gentleman over here, and there was a lady at the back. Yeah, okay.
Yeah, thank you. I wonder if you have considered extranuclear non-genomic actions of estrogens and androgens, because I don't think this is measured with this test.
Yes, I do.
And this is half, at least, of the
So you mean the
phosphorylation of ERK, MAP kinases, and other effects that are
exactly,
there.
But this is the non-receptor binding effect.
No, it is receptor binding, but it's the receptor outside the nucleus.
Yeah, no.
And this is more than half of the signal that these receptors produce.
Yeah.
And it is known for 20 years. And
So
I think
so we are doing the steroidogenesis. for the synthesis of our hormones, but it's not with the HPTLC. So what the HPTLC is a solution to identify, but it doesn't mean that we are not assessing the other bioassays. You know,
Yeah,
but for what you are mentioning, we are not applying yet. But
yeah, so
this
is something to incorporate to the bioassays, I think.
Yeah, thanks for the tip.
Okay, and one last one at the back, and then we'll wrap up.
Thanks a lot for this very interesting talk, Lisa Zimmermann. My question is, did you or are you currently considering also applying assays to other endpoints to explore other endpoints, like for example, such endpoints which are mentioned in the chemical strategy for sustainability, like specific target organ toxicity, neurotoxicity, things like that, or?
This is This is, we are developing, we are working on the thyroid area because this is very, very important on the development, but with the organoids. We are developing the platform for the organoids because this is very key to develop a methodology for the thyroid effect. And there is one paper that they tried with the HPTLC with the thyroid, but I haven't tried. But yes, we are trying to, we would like to explore other,
Perfect. Thank you so much for sharing those views, Maricel.
Yeah.